We should be live. All right. So, welcome everyone to the next workshop of Blueprint, our second workshop of BP week, as we, excitedly await for this Saturday and Sunday for Blueprint, the hackathon itself to happen. But thank you guys all for coming. And today we have an awesome workshop from MIT BioMakers, who are excited to, share a lot of their insight and just to provide a lot of their experience to you guys. So, without further ado, I'll hand it off to them.
Hey guys, I'm Leona. Give me a second to share my screen. Okay, should be working. So today Maya and I are going to be presenting a little bit about MIT BioMakers. Also, let me know if everything is good with the screen share.
Yep, all is good.
Okay. All right. So we're going to do a workshop on two different computational biology techniques, and we're going to tell you guys a little bit about MIT BioMakers. So teaching today's workshop, my name is Leona. I'm one of the lab co-coordinators for MIT BioMakers, and Maya, she'll introduce herself later, is also a lab co-coordinator with me. We're a student group who works around this space called the Huang Hobbs Biomaker Space, and we organize community events. We work on different workshops. We teach people about biology techniques, and we even do community projects. So wet lab research together as a team. Biology research requires a lot of resources, and so we're really lucky to have this space. It helps make bioengineering an engineering discipline that's accessible, like electrical engineering and mechanical engineering. So we can have this sort of playground for biology where we can play around, we can have the reagents, we can have the special equipment that's needed to do independent projects. What's really special about this space is that it's entirely for students. So we have all of this equipment, and people can put in a project proposal and then they can do the work in there. So some former student projects were something called Remora, which is a blood platelet-aided drug delivery platform. Sanviato, which was a way to detect blood clots, and then Skin Morph, which was a second skin layer that changes its properties. And then this is one of the workshops we run using engineered E. coli to make sort of bioprinted art. Okay, so my workshop today is going to be on modeling biological systems with differential equations. I'll explain what that is since I know you guys are in high school, and MATLAB, which is a programming kind of, we can think of it as like a grapher for us, and a differential equations calculator. So the first thing that I'm going to teach is what is a derivative. So a derivative tells you the instantaneous rate of change at a point. And even if you guys haven't taken calculus, like I think this is a concept that everyone can kind of wrap their mind around as long as you understand what a slope is. So if you remember from algebra, slope is rise over run. So in this line, the slope would just be m. I'm not going to tell you guys how you actually compute a derivative. There are a bunch of rules that you learn when you do take calculus on how to actually do this. But since MATLAB is going to do all of the computations for us, you just need to conceptually understand what this is doing. So here, I'll teach you a little bit about the notation. Y equals mx plus b is our equation, and then dy dx means like a little bit of y divided by a little bit of x, a little bit of rise over a little bit of run, and that's the derivative, and that is m, which would be our constant. So if it's like y equals 5x plus 2, the derivative would be 5 because the slope is 5 everywhere on the line. So some people like to have a physics kind of view of things to help wrap their mind around something, also because it's in a real world kind of example. So imagine I throw a ball in the air at a certain time t. Y of t is like our function of how the height of the ball is changing at each specific time. Y is the position, the height at time t. Dy dt would be the velocity of the ball at time t. And the derivative represents the velocity. And that's the rate at which the height is changing. So it's maybe going up faster at one point than another point because gravity causes acceleration, or going down faster than at another point. And so d squared y dt is just the notation for a second derivative, which would be the acceleration at time t. A second derivative is when you take a derivative of a derivative. Okay, so what if instead of a straight line, we have like something curved, some kind of parabola? And this could be moved up and down, whatever. If you have a certain point on that parabola and you wanted to find the slope of that, we would take the derivative. And then if we wanted to find that slope at a certain point, let's say it was at x equals 5, the slope at that point, we would plug in 5 into our formula we computed for the derivative. Okay, so conceptually, that's pretty much all you need to understand about derivatives. It's kind of how you measure instantaneous rate of change at a certain point. And this gets way more complicated because you can have functions of more than one variable. You can have multivariable functions. And so it can be like this kind of landscape where instead of it just being a tangent line where you find the slope of it, it can be a plane, or even in higher dimensions that we can't even visualize. But what's nice is that we have MATLAB, which is kind of like our calculator. So don't worry about all of that. So now I'm going to explain the concept of a differential equation. A differential equation is an equation that's describing how quantities are changing in relation to each other. And so these are really vital for science and engineering. You can have circuits in electrical engineering, like you can imagine flow of electrons. You can also have circuits in biological engineering, which is what we're doing today. You can imagine a spring in mechanical engineering, and how that spring is oscillating can be modeled as a differential equation. And so a really simple example would be dy dt equals k times y. I should have written as k times y of t, but it's basically saying that if you have y, some function y that's in terms of time, and it's changing over time, how much it's changing over time is proportional to how much of y you already have. And the proportionality constant is this constant I put in called k. And this describes exponential growth. So this is relevant to so many different things. It's relevant to population growth, it's relevant to bank interest, etc. And solving a differential equation means finding the function y that satisfies the relationship. Okay, so now I'm going to do a quick bio review so that we can understand what we're doing today. I want to make sure this is accessible for everyone, no matter how much bio you've taken. But basically, you know that our cells have DNA in them. DNA is like our code for life. And that gets transcribed into something called RNA, which is kind of like the intermediate code. And then that gets translated into proteins. And proteins are made up of a bunch of amino acids. What else should I explain? Let's see. So DNA is transcribed into RNA, and it has a certain promoter region on the DNA where things need to bind, different factors and things in the cell. Don't need to know all the details that cause it to get transcribed. And then RNA is translated into proteins by ribosomes. And so RNA on it has something called a ribosome binding site, and it can have different strengths. So a stronger ribosome binding site means that it's more likely to get bound to, or it gets bound faster. Like you're going to get more protein out of that RNA transcript. And then I'm also put in the term expression because sometimes I say expression just to mean like a gene getting expressed. A gene is like a segment of DNA. And that just means its proteins are being made. And then repression is like the opposite of expression. Repression is when something maybe binds the DNA and blocks it from being able to be transcribed to RNA, and then its proteins don't end up being made. So why are differential equations powerful for biology? Well, biology, like a lot of other things in the real world and science, is all about change. So, growth and death and gene expression and protein degradation and differential equations are designed to model change. So, it can model all of these things. These dynamic things are also often related to each other. Um, and so, if we want to maybe develop a new drug or we want to design a gene circuit, we want to understand how the different parts of the system are related to each other and how their quantities or concentrations are changing, or if you maybe change one thing or one thing mutates, how it's going to affect the other things. So, differential equations let us build a mathematical representation of these processes and predict what will happen under new conditions. Um, in synthetic biology and systems biology, ODE models guide the rational design of synthetic gene circuits and enable prediction of a system behavior before building anything in the lab, because lab experiments are costly and take resources. So also lots of projects, say in the biomaker space, you might want to kind of figure out a model for what's going to happen, or do some math equations to figure out what concentration of something you need to mix up. Also, I'm going to have a little sidebar. So I'm doing the uh, theoretical kind of side of computational biology. So you start with the theory and the science and the math and build a model based on that. And Maya is going to be doing the start with a bunch of data, and then you analyze that and make some conclusions about biology. So these are two sides of computational biology. Both are really important. The side of like collect lots of data and then analyze is kind of where machine learning and stuff lives. And then this side of things is like right from the theory and the synthetic the differential equations. It's actually very inspired by electrical and mechanical engineering. It's very much from physical principles. Okay, so I've been saying the word synthetic biology a lot. I actually should define that. Synthetic biology is this relatively new field where we're essentially building biological systems from the really basic building blocks, like from genes and uh, and proteins, and we can also have cell circuits where different cells are interacting with each other. And so we have these different parts that we have, these basic building blocks that over time researchers have characterized. So we have the certain gene that maybe outputs a protein that's fluorescent, so we can monitor it, and then and so we can see like how much of that is being produced. Or we can have a certain gene that's repressing another gene, so it means it binds to the other gene at the beginning and prevents that gene from being expressed. And so maybe if that other gene put out a different color fluorescent protein, we could see that color go away, and we could see that's being repressed. And so here's just like a cool, this is a pretty famous, I think, cancer model, just like of a bunch of different pathways in the cell. And you can see how things get really complicated in biology. And if you have a mathematical model, it can be really helpful for understanding these systems. So some real applications are circuit design, drug target identification to identify like which proteins or genes could be disrupted to treat a disease, pharmacokinetics and dynamics for how drugs are absorbed and distributed throughout the body, and metabolic engineering. So how we can engineer microbes that produce biofuels or drugs or industrial chemicals, which I think is really, really cool. So the big challenge is that modeling biological systems is hard because like these are so complicated, these systems, that to model it, you need to understand a lot about it. So they're actually, you have to do a lot of research, a lot of data collection to understand the different interacting parts. And so even if you know the structure of the interactions, you need numerical values for rates, which you need to experimentally determine. There's a lot of incomplete knowledge, and then it's difficult to do that data collection in vivo, which means in living systems. And then, of course, once you get the data, it's noisy. But so for today, we're going based off of a really nice ODE model from a foundational synthetic biology paper. This is the paper. I can send out the link after this. It was published in 2000, and it helped launch the field of synthetic biology. It's called a toggle switch, You can imagine kind of like an on and off switch. So this is a nice schematic of the toggle switch. Essentially, you have two genes that are like mutually inhibiting or repressing each other, and they have inducer inputs, which is going to be like a molecule that you add. And two promoters, which, like I mentioned, are kind of the part that's regulating the gene it's paired with. And this system has something special called stability, which means that it stays in a stable state even after the initial chemical signal is removed. Okay, so this is a model, really, really clean, just two differential equations. So we have an equation for U, which is the concentration of repressor 1. Their repressor they used was called LAC-I. And then V is the concentration of repressor 2, which was something called CI. And so you can see how we're relating U, which is the concentration, to its own derivative. And you can see how we're relating V to its own derivative. Then alpha 1 and alpha 2 are the effective maximal production rates of each repressor. And beta and gamma are the cooperativity Hill coefficients of repressor binding to DNA. I'll cover what cooperativity is. So cooperativity is this idea of how if multiple things are kind of working together, it can affect the effectiveness of the repression. So you can imagine, so this is about the cooperativity of the inhibitors binding to the genes, the gene promoters. So imagine you're trying to block a doorway. You're trying to block the expression of this gene. If you stand alone, someone can push past you and then transcribe that gene. But if you and then your friend also comes and stands next to you, it's easier to block that door, and less is going to get past you. And your repression curve is going to become sharper. And so that's the idea of cooperativity. Like multiple copies of a repressor protein can bind together as a dimer, trimer, tetramer, which is just 2, 3, or 4, and they're more effective as a team. So mathematically, there's something called the Hill function, 1 over 1 plus x to the n. When n equals 1, that means there's no cooperativity, and the repression curve will be a gradual, hyperbolic, kind of gentle slope. And when n equals 2 or higher, that's cooperative binding. And so the curve becomes steeper and sigmoidal, which just means it kind of looks like an S, and it's like a sharp step kind of function. And that steepness is what creates two distinct stable states. So then a little bit about MATLAB. I don't know if you guys have used MATLAB before, but I'll keep it very graph heavy, coding minimal. I'm not just going to quickly look at the code. But it's a very powerful tool for solving differential equations and modeling systems. It has this solver called ODE45 that we're going to call. It uses the Runge-Kutta method. I don't know if I pronounced that right, but you guys don't even know what that is. Just think of it like a nice calculator that does the work for you. And we can easily also tweak parameters. And so I'll send out like the code and you guys can play with it. And so Gardner and Collins used the exact model that we're going to use in the MATLAB to predict which circuit designs would produce a working toggle switch, and then they built them and confirmed the predictions in the lab. Okay, so I'm now going to go into MATLAB. Let's go to the beginning. So if you've never seen MATLAB before, this is what your screen will look like. You can install it on your computer. You can open the file. So I would use like this and then go into your downloads and open it up. And then it'll open up here. It's a .m MATLAB file. And let's see. So here is your workspace. You can think of this as kind of like a nice tracker for all of your variables that you have in your code. So each time you create a variable, you'll see it over here in the workspace. MATLAB is also very powerful for matrices. So I don't know if you've done matrices in math class before. Basically, just think of it like a giant table. and it can have multiple dimensions. You can also think of it like a anyway, it's a big array of lots of numbers, and you can manipulate them, and it's very useful for storing values. Um, and yeah, we have people with all levels of programming experience, have been told, but yeah, it's what we're doing sort of in this code is we can uh, set values for different variables. We can print things to the command window. So I have this, oops, this output from the code that just summarizes what we got for the parameters that we set. And we can make nice graphs. So there's syntax that you can search up on the internet for what you need to pass into a command to make a graph, like what you want the x-axis and y-axis to be, what kind of graph you want it to be, what colors you want things to be, labels for the axes, that sort of thing. Okay, so I've split this up into a number of sections. In the first section, we just define our parameters. So we set alpha 1 and alpha 2 both to 10, and beta and gamma both to 2. 2 is greater than 1, so they're cooperative. And then these are some production rates. As long as these are balanced, I think it works. So you could play around with it. You could set them both to like 6. But also you could try setting like alpha 1 to 2 and alpha 2 to 10, and then I think it wouldn't work because two of the main conclusions were that you need both cooperativity, so Hill coefficient greater than 1, and balanced alphas for the switch to work and be bistable. Okay, so section 2, we're going to do null-cline plots. So a null-cline is the set of points where one variable's rate of change is 0. So here we're just kind of setting things up. We're finding these null-clines where du dt is 0 and du v dt is 0. And then we do a plot, and I'll open that plot up. Okay, so we can see here that with our parameters, the null-clines intersect at 3 points. There are 2 stable steady states. So here's a steady state, and here's a steady state where they intersect. I would like to actually maybe shift this graph a little bit because it's hard to see. But yeah, that's where both rates of change are 0, meaning the system is a steady state. And then we also have one unstable point in the middle. And you can think of this as like repressor 1 winning and repressor 2 winning. You can see on the axes, this is the concentration of V, and this is the concentration of U on the x-axis. And so this is how a synthetic biologist could design a circuit computationally or assess different parameters for a circuit before actually going to the lab and building it. Because by plotting these null-clines, you can observe if your circuit's going to have these stable states. Okay, so section 3, these graphs got a little bit messed up because I was messing with them before this, but I'll just explain sort of the general concept of it. We want to simulate the toggle switch from different initial conditions and see what different states it settles into. So we're starting in with different initial conditions. So remember I'm calling these basins, but remember this point on the far sides and then the point in the middle. So we're testing what the quote unquote trajectories of those are, and the trajectory is just what steady state it ends up, towards which steady state ends up going. And we use ODE 45, which is the MATLAB ODE solver. Here we're solving, and then we do a plot, and we have this. Okay, so a little screwed up, and this one's also a little screwed up, but the general idea here is you can see how the protein concentration goes to these 2 different levels. And in the phase portrait, you can see how it goes to 2 steady states. So, but yeah, don't look at these. The concept, I guess you can liken to a landscape with 2 valleys and a hill in the middle. So if you were to like place a marble somewhere in the landscape, it'll roll downhill and settle into a valley. And so the valley it ends up in depends on which side of the hill you started from. And so theoretically, if you were at that middle point, you put down a marble, it wouldn't roll to either side, but in a biological system, you're going to get a perturbation and it will roll to one side with like the slightest nudge. We're plotting these 2 quantities against each other. And each point on that graph on the right was every possible state of the circuit with each combination of U and V. But yeah, so that's like the main idea of bistability, which is that with the same circuit and the same DNA and the same equations, you can get cell memory. So depending on what the cell's initial condition was and where it started, it can be in a completely different state, which is really cool. Okay. And yeah, you can think about like what important uses of cell memory could be. So if you have like contaminated water and you engineer bacteria to be kind of like a sensor with this cellular memory, if it ever it encounters a contaminant, it could have this bistable switch that could be its inducer, and then you could have like a living biosensor. That's just one application that I've heard. Okay. So then section 4, I really like this one. I think this is a nice illustration of it. In the real experiment, they used IPTG or heat to flip the switch as an inducer. And we're simulating this in MATLAB by just increasing production of one repressor, which is going to overwhelm the other and flip the state. So let's go into, yeah, there's nothing else important to explain there about the graphing. Okay, so our first graph, we have protein concentration, and the blue line is repressor 1, and the red line is repressor 2. So the system starts out in this state with repressor 1 at a higher level and repressor 2 at a very low level. And then we're going to say, and this is the inducer signal. So at t equals 25, right here, we add inducer, which temporarily is going to boost the production of repressor 2. And then it's going to overwhelm repressor 1, and then the system flips to state 2. And then when the inducer is removed here at t equals 35, the system is going to stay in state 2, which is really cool. Like the switch gets flipped, and then it actually stays here at the end. You can see in the opposite states. So you can see how it started out with blue on top and red on the bottom, and it ends here with red on top and blue on the bottom. Okay, and getting up to the end of the time, so I'll go quickly through the last two graphs. So, this section is going to talk a bit more about cooperativity and its effect. So we're looking at different Hill coefficient, which remember that was the n. N equals 1 means no cooperativity. N equals 2 means cooperativity. So we're going to look at different Hill coefficient n values, and we're going to see how that affects our plots. So yeah. Okay, so you can see how here there's only one intersection point if n equals 1, and it's what we call monostable. And we're not going to get that bistable effect that works for a switch. Here at 1.5, it's kind of near the threshold. I actually should have adjusted the parameters a little bit more because you can see that there are two intersections. But it should be like, if it's right at the threshold, it should be like just kind of tangent to each other. And then here, we've got bistability, and then here, see how it gets just more pronounced how it's bistable, and the graphs are more separated. Okay. And yeah, that's an important design principle, which is that cooperative binding was required for the switch. And so that's why the team in this paper chose repressors that were known to bind as dimers or tetramers. Again, dimer, two things binding together, and tetramers, three things binding together. And finally, remember how at the beginning I said that the production values need to be balanced for the switch to work, the alpha values. So we had alpha equals 10 and alpha equals 10, that works. But if we had like different values like 2 and 8, that would not work. So we're just plotting Uh, Here's an example with balanced. So if both are 10, and here's a very imbalanced example. Here's another imbalanced example. And you can, um, and it's just flipping the two. And you can see how the balanced example is the one that works. Again, the axes need to be changed a little bit for this. You guys can also play around with the plotting when I share the script. These are just done pretty quickly. But yeah, when the production rates are balanced, the null lines will cross 3 times. Um, and it works. So in the real experiment, their team tested different ribosome binding site variants. As you guys remember, ribosome binding site was what affects how much the ribosome is going to bind and how much protein it's going to make. So it's basically tuning that strength. We have a bunch of different characterized ribosome binding sites that we can engineer in and choose. And then we can basically tune the expression levels to what we want. Um, and so they found a construct that worked exactly like the model predicted. So yeah, mathematical modeling can guide real experimental design. And then yeah, you can play around with the parameters in here, and you can just here's your command window, and you get your output of like just kind of some summary stuff. And yeah, so I hope that that was a helpful kind of overview of how MATLAB can be a very powerful tool. And I'll hand it over to Maya now.
Thank you so much, Sienna. Okay, buckle up. I have a great workshop also prepared for you guys. Okay, as long as I share screen. Okay, can everyone, I'm hoping all the slides can be seen. Okay, I think
I can see
so.
it.
Okay, so we're gonna go ahead and get started. As you know, my name's Maya. I'm a co-lab coordinator with Leona, and today, as she perfectly prefaced, we're gonna go into more of like how do you design programs to answer a biological question. And we won't be using MATLAB, we'll be using Python. But without further ado, let's get started. Okay, just into a bit of a brief background about me. I was in your shoes once to all those Blueprint people that are streaming this right now. I participated in Smath Hacks 2024, which is a hackathon in North Carolina. And then the following year, I was on leadership team for that hackathon. And then a similar trend happened for Hack MIT. I was accepted to participate in the hackathon of 2025, and I ended up joining the team. And the rest is history. But what did I even do in high school that introduced me to programming? I really enjoyed math. And when I was able to take classes at college level, I ended up taking some discrete math classes and an introduction to Python course. And that combined with motivation and passion and just excitement also allowed me to participate in a summer program in data science. And then before starting here, after I graduated my senior year, I participated in an internship that's a collaboration between the Air Force Research Labs and Wright State University and the University of Dayton. It was the Sensors Directorate Internship Program. And now I'm here today. So I'm excited to show you guys how um, anything intersecting with math and programming can be used to answer questions about biology. So, what my goals are for you, you'll be seeing program in action towards the end of the workshop. You'll go ahead and learn how computer science and math intersect to answer biological questions. I'm hoping that you'll learn some skills that can be used at Blueprint, which is coming up this weekend. And then I'm also hoping you'll take some of these skills and see how they can be used beyond Blueprint. And at the end of my presentation, we'll have a question and answer session, so you can ask us anything with regard to our presentations today. And I hope you have fun. This should be fun and really accessible to anybody, regardless of their programming level. Okay, so a little bit of background you should know is that for biological problems, they require more than just biologists to solve them. And this is in context to the research I do. So I, for example, work at the Manalis lab here at MIT, and the Manalis lab is really, really good at developing essentially small devices called microfluidic chips to study single cells. And this couldn't be done without people that are experienced in mechanical engineering, people that are experienced in chemical engineering, and people that are experienced in computer science. And holistically, we can see this beyond the Manalis lab itself. The Koch Institute, which is where my lab is housed, it brings together cancer researchers with people who build technology. And the point is that science needs all kinds of thinkers, and that includes people like you. But to preface a little bit more of the biological specific background of this workshop, you'll need to know a little bit about what a T cell is, a little bit beyond what that it just exists. T cell is a type of immune cell in your body. So you know that whenever you get sick, there's a response happening inside of you, and T cells are like your immune system's soldiers, to create an analogy. On the left, you have a cytotoxic T cell, and these help identify and kill infected cells. And on the right, you'll see a B cell. And B cells are really good at releasing antibodies that say stop a virus from entering your cell in the first place. So, understanding how these cells move around is what will be the basis of this workshop. And intuitively, this also will inform scientists about how our immune system works and how we might be able to help it work better. So, a preview of the technical side of the workshop. You'll be learning what GitHub is if you don't already. You'll know that Fiji, and Fiji is just ImageJ, is the program we'll be using that houses the plugin for the workshop, which is called TrackMate. I'll be showing you a collab notebook, maybe two specifically if we have time, and then a preview of some tasks in that those notebooks are about visualizing data and reducing high dimensional data. And I'll give a background for three techniques specifically, and then we'll wrap up the workshop. Okay, so the prep that I did beforehand, because we don't have time necessarily for you to also go alongside and do it yourselves, but what I do have is the link to the GitHub and the data, but I already had Fiji installed, which I used for my work in the lab, and I downloaded the data set and also the parameters for TrackMate. And this has a big batch of files, 82 to be specific, but I'll show you the notebook, and within the notebook itself is a section to download the test data set, which is a little bit smaller. Um, but those are the two different pieces of data that are both can be used in the workshop. So the tools we're going to be using today are TrackMate, as you've heard me say, and then CellTrack's Colab. These were both developed by researchers at the Cell Migration Lab. TrackMate, as I said, is a plugin for Fiji, and it helps you track particles and cells in microscopy videos. It doesn't have to be limited to just cells. For example, a particle could be a bead, and what I do for my research is I'm actually tracking beads. And then CellTrack's Colab takes in data from TrackMate and lets you have a bit of a deeper analysis and visualize the tracks that you generated. You can think of it like this, that TrackMate watches the cells and moves and records their positions, and then CellTrack's Colab is what helps you make sense of that data. Okay, so a little bit more about what TrackMate is doing. You don't need to know the nitty-gritty of it, but you can know that it's free, it's open source, and it works inside Fiji. There's plenty of documentation that dives a little bit more into how it works and what parameters of it are most useful for your data. But what it does is that between frames of a video, it'll connect dots, and those dots are called a spot, and then it'll link them, and that's called a track. And it's kind of like a path the cell or particle you're looking at takes over time. It has a nice step-by-step interface so that if you don't get to see it all in action today, you can go through it pretty much by yourself independently. Okay. And then what is CellTrack's Colab? CellTrack's Colab is a companion tool that runs inside Google Colab. So you don't have to download anything except the dataset, but you open Colab inside your browser and it runs. And it takes the tracking data from TrackMate and you can compile it, explore it, filter it, visualize it, and it's designed to be very accessible. So you'll see lots of comments and markdowns about how to go through the notebook and analyze your data. Okay, so a little bit more about particle and object tracking. This is a quick breakdown of what it actually means is particles usually have a center point, and then that's called a centroid, and then the object tracking is more detailed because you're eventually drawing like a boundary. You can imagine that as like circular or elliptical if it's a boundary over a cell. Or for example, on the right, you can see tracking with the example of drawing a rectangular box around a person. But you can see that this just isn't limited to biology. You can think about applying these concepts to tracking things like cars on a street. And the math behind this is all similar. But specifically, the applications in cell biology that we'll be looking at today are how scientists use tracking. They can use tracking to individual proteins inside cells. We'll be studying just how individual cells move and migrate and move around today. But you can also study how organelles within a cell move. And organelles are just tiny structures, like your nucleus, a chloroplast, or a mitochondria, and you can measure how these things even change shape over time. The picture on the right is from a paper in TrackMate published in Nature, and it just shows you can see a collection of cells and the tracks are overlaid onto them. You can see it in motion too. Okay. Focusing on the tracking principle in TrackMate, this is the basic workflow, input segmentation, and object linking. And what is segmentation? Is what we'll be focusing on segmentation and object linking. Segmentation is the process of identifying and separating individual objects in an image. And there are a couple different methods to use it. You can use like deep learning models, for example, for segmentation. You can use models like CellPost or Stardist or just a couple on the left. And these are pre-trained neural networks that have gotten really good at recognizing cells. And TrackMate integrates basically all of these combined, and that's what makes it really powerful and flexible. But you don't need to know the nitty-gritty of that. What you should know about segmentation is that it is getting an outline of each cell, and it's less important to see how the cell is counted on its own. The main problems are that we don't want over segmentation, which is when one cell accidentally gets split into multiple objects, but we also don't want under segmentation, which is where two cells get counted as one. So both of these would mess up tracking data. So getting this step right matters a lot. But TrackMate's, the plugin's already gotten good at optimizing how you can segment your data. Okay, the second thing we need to think about is TrackMate object distinction. So there are a couple different methods. I personally use the log detector because it's good at detecting spherical things. Cells are usually spherical, and then beads are usually spherical. But what you can do is that you can filter using different detectors based off of how circular or blobby shapes your objects are. And what's also important is that sometimes if you want to, you can apply a label or mask over your images using other tools. What's really cool is that TrackMate gives you this diversity of a menu to pick which detector works best for your data, and that's what you should keep in mind. Okay. The next thing we're going to be looking at today is object linking. So this is where the actual tracking happens. It's after segmentation, where all the cells are in each frame, that a tracker algorithm figures out what cell in frame one corresponds to which cell in frame two, and so forth. And it's these tracking algorithms that are the pieces of math that kind of make smart guesses about which objects are the same types of cells across time. And then I'll show you two different examples. So I use the LAP tracker. This stands for linear assignment problem. It's very simple, so you can define this linking distance, but essentially every cell gets a search radius around it, and if a cell in the next frame falls within that radius, the two get linked as the same cell. It's basically asking what's the most efficient way to connect all the dots between frames while minimizing total movement. And that's the like assignment part of it. It's a classic mathematical optimization problem. There's another tracker that you should be aware of, and there are definitely more in documentation, but this is just another important one I think you should know. It's a little bit more sophisticated than the LAP tracker, and instead of looking at where a cell is right now, it kind of uses past positions to predict where it will be. If the cell is moving unpredictably, the Kalman filter also accounts for that uncertainty too. So it then links the prediction to the closest actual measured position. And this works really well for cells that are moving consistently in one direction. Okay, that's tracking. What if you have multiple movies to track? And this is where TrackMate Batcher really comes into play. This is a real world example I do with this. When you're a biologist, you do multiple different replicates of an experiment with multiple different conditions, and maybe even multiple different days to get day-to-day variability. So what TrackMate Batcher can do is you set your tracking settings once, save them as a file, and then drag and drop all your videos in to TrackMate Batcher, you hit run and it processes everything automatically. And then it exports the data you want. You can check this off in the export settings right here. This is exactly how you'll see it should be on your end if you look at it in the future. But you can export spot tables, track tables, and even tracking movies for each video. And this is a big deal in research, especially for biologists. Okay, getting into the question we're answering today, we're answering the question of how does coating on a glass dish affect how T cells, which I mentioned in the introduction, how they migrate. There are two different conditions, so you don't need to know too much about except that they're two different adhesion molecules. So one dish is coated with intercellular adhesion molecule 1, and the other is coated with vascular cell adhesion molecule 1. And so these are just two different proteins that are naturally expressed. And by comparing how T cells move on each surface, we can learn something about how those proteins influence immune cell behavior. Okay, so this is the overview of the tracking analysis. Once you've done all your tracking, you're left with a lot of data, multiple conditions, multiple experiment repeats, multiple fields of view on a microscope, and lots of files. So this is where things get overwhelming if you do it manually, and that's why CellTrack's Colab exists. It makes it manageable to analyze all your data in one place so you don't explode. So we're going to dive into a tutorial in Colab, and I'll give you a bit of a preview about what we're going to be doing. Um, there are multiple different notebooks. We're going to be looking at one, which is visualizing data. That's the CellTrack's Colab TrackMate. If we have time, I want to show you how dimensionality reduction works. So just to preface, I've already explained this, but we're using Google Colab, we're loading in Python, it loads into a Jupyter notebook where you can run cells modularly, so run one cell at a time. And this is the workflow, which I've already discussed. So I'll show you a little bit about what the setup of the data looks like before I open the notebook. So these are how the folders in the test data set are organized. And this is all that CellTrack's Colab requires to run. So even if you weren't using this specific test data set, this organization could be used for data and then loaded into the notebook. Okay, just a preview for what you can, what the power of this notebook is. You can visualize tracks and superimpose them onto a plot. And that's just what each of these three graphs are doing. You can even plot a vector. Leona mentioned earlier what a derivative is, and that's slope. A derivative of position over time is speed. And a vector is just applying a direction to the speed at which something is moving in, and that's what the right plot is. You can apply some filtering to your data, and this is what where data quality comes in. If you don't want to look at all your tracks at one place, you can filter by certain parameters, like tracks that have a slow speed or a fast speed. If you know all your particles should be moving, you can filter out distances of 0 and filter for the distance you expect your particles to be moving at, and so forth. Some features of the tracks, and this is where dimensionality reduction can come in the future, is that this is the output of the tracks. There's a bunch of useful features, but all that you need to know is that these numbers can let you compare cells across different conditions, and then by having all these parameters, like how fast a speed is, how straight or curvy the path is, these are just different pieces of information you can get from a track output. For the spots themselves, in addition to the track level, you can get down to like individual object detection level parameters. So these come from the spots table that TrackMate outputs, and things like cell intensity, that's like how bright something is, metrics like shape or circularity, like aspect ratio. Um, these level of details can like tell you if maybe a cell changed shape as it moved over time. Um, but as you can see, like each of these parameters has a data point, and that is a lot of data in the end. So if we get to it, I'll show you some techniques for dimensionality reduction. Okay, so at the end of the day, I want you to know like what you can do with this data. Um, the big picture is that a lot is possible with this data. You can do quality control to make sure your data set's balanced or unbiased. You can look at subpopulations. Maybe some cells are moving fast and others are barely moving at all. If you're interested in more of the biological questions, you can run statistical tests to see if the differences between each condition are real or not. And all of these are possible, and these techniques aren't just unique to working with biological data. The concept of controlling for quality data, balancing an unbalanced data set, looking at trends, reducing high dimensional data, this is useful in a lot of different projects. Okay, quality control, which I think you should know a little bit more about for your projects in the future, it's basically looking at like the weight of each repeat and looking at if they're equal. So about as you can see on the right, the data sets are not balanced. You can see that the like ICAM condition and the VCAM condition have a different number of data points for those tracks in each of the repeats 1, 2, and 3. So what one thing you can do is you can look in subsample populations to eventually get a data set where the number for each condition is equal. And as I said, this isn't just unique to working with a specific data set. Data sets are commonly not balanced. So this is important to get a grasp on in the future if you want to use this for a project. Working with high dimensional data, I'll give you a preview into the theory and you might get to see the practice. But I'll give you a simple example of a linear technique, which is principal component analysis, and also a technique of reading documentation. So you don't have to know immediately what all these things do, but you should have an intuition, and go look at documentation that can inform you of what you're doing before you do it. So for principal component analysis, for example, it's a way of essentially projecting a lot of dimensions in a matrix into a linear form that you can understand. Like a graph is usually an XY, and that's where you can see with the graph of principal component 1 and principal component 2 of this random data set where someone also used this common technique. And it's just a way of being able to make sense of your data because you can't really make sense of data that might be in 5 or 20 dimensions. But what you can do is you can reduce it to 2 components. Um, there are multiple different methods of doing dimension reduction. There are linear and nonlinear ways. You'll see a nonlinear way in practice in the notebook, but I encourage you to explore the documentation on each throughout your projects. The 2 examples I mentioned that'll be in the notebook are UMAP, TSNE, and HDB scan clustering data. And these are like example plots pulled from the notebook. And all you need to know is that they're different from PCA because they're nonlinear methods of reducing high dimensional data. Okay. So the takeaways from you at this time should just be that TrackMate and CellTracks provide powerful tools for analyzing cell behavior and tracking particles in from large amounts of biological data. And more broadly, documentation's your friend. It's okay to be a beginner. And then projects on GitHub and public datasets, like for example on Kaggle, can help you explore ideas. And I hope at this point you see that programming is relevant for life sciences and all fields in general. Okay, but right now I'm going to stop sharing the PowerPoint and we're going to move over to the TrackMate notebook, which you can hopefully see at this time. Um, and I'll just walk you through it. I've run most of the cells so that you can see the plots since the dataset takes a bit of time to download. But these are essentially how the experiments are organized in the folder. And then the this is preparing the session, mounting Google Drive since Google like Colab runs data with on Google Drive. You don't have to say download a data set locally onto your computer. And then this is importing every library that's used in the session. And as I said, we don't have time to go through each of these libraries, but they each have documentation. And so we'll just go down to, this is still importing the data. I already have it downloaded, so I just loaded it from my folder, my Google Drive. And these are examples of what was recorded in each column. For example, you have the ID and the track ID. You have the shape index, number of gaps in the track, number of spots, the total distance traveled, and these are all what comprise the data set, which is the output from TrackMate. And we'll go in to visualizing some of the tracks. So, for example, here's like ICAM condition 2, repeat 1. This is a coordination. Oh, stop share. Share again. I think my notebook wasn't sharing, but you should be able to see the notebook now. Great. Okay. Um, okay. So what you can see here is you can see the coordinates for one of the conditions, ICAM repeat 1, experiment 1. And you can like superimpose the position of the plots onto a onto a graph. That's what I'm doing here. And when you can do that for multiple multiple points of data, multiple conditions. And that's all this is doing. And as you can see, all the tracks are different. And so you can get a visual sense of that instead of looking at all of that in a CSV. Okay. And these are all the same plot. So we're going to go down to some of the other plots. You can, well, first, this is a drop down. I chose the condition I wanted to look at. And then what you can do is you can normalize them. What normalizing is doing is that not all, you might be having like a disproportionate or like not representative form of a track if, say, one condition already has super short tracks and they're not as long as the tracks recorded from another condition. So you can normalize that and then get this plot, for example. And normalizing, you can normalize by anything. If you're looking at how much protein synthesized by a cell, you can normalize by the size of that cell and so forth. So this has multiple different applications. Um, these are processing multiple different fields of view. And if you want to, you can edit all of this. This has code. And you can just like edit the scale if you would like. You can press show code on everything. These are more plots for each condition. And like I said before, this is one of the examples of the vectors you can get. If you take each track and apply a direction of which way the cell is moving, you can plot all those vectors onto a map like this. Okay, the data quality, you can filter tracks by certain parameters. And here I have a way you could just you can just drag the parameters if you want. Um, I've already filtered out the tracks that we're going to be looking at. And here, this is a plot of like the original data compared to the filtered and smooth tracks. And that comparison you can get for any condition. And we'll just keep going through the data set. So this has a couple more like tips and other things you can do. You can calculate duration and speed metrics for your data. You can do like directionality. If you know what Euclidean distance is, like straight line distance is what this is computing, and that is all that's in the remaining of the notebook. Okay. Since we're at 9, I'm gonna go ahead and hop over to the dimensionality reduction. and stop share and go back here. Okay. We'll start at top. Okay. So what this is doing is that it's doing the same thing of loading the same original data set, visualizing the same tracks. And what you can do is you can explore your data using UMAP and HDBC scan. And these are just two different methods that you can use. You can use other dimensionality reduction techniques, and these are just two examples that are in this notebook. There's a little section, a blurb about what UMAP does, and I would have to run all these cells from the beginning if you want to see this. But what it does is I can go back to the PowerPoint and show you what like an example output should be. Okay. And what was it from current slide? So, like I said, these two, when you run that UMAP cell, I would have to run all the cells in the beginning, which would take probably about 10 minutes from what I've estimated. But you can get these two examples of plots of reducing all the dimensions from all the parameters I showed at the beginning to more like two that you can make sense of in a figure. Okay. And these notebooks are available on GitHub. You can explore more of the advanced or like beginner methods as you wish, but at probably going to wrap up the workshop soon and get to some questions. Okay. So I wanted to explain what this means for you about Blueprint and how you think about projects and execute projects before we get into questions. So what you should keep in mind, this is what I want you to do throughout the hackathon and the learnathon. You can think about what biological or health problems you're trying to solve. You can think about a target audience, for example, like me as a researcher, I use Cell Collab. You can think about what you're designing. Do you imagine a researcher, a healthcare professional, or a patient using what you design? What impact you're trying to make. For example, here we're trying to answer a biological question, but you could design, for example, an app that instead is used by a patient. You can think about what tools already exist and how you can do something different on a smaller scale. And TrackMate and Fiji are just like one of the tools. You can think about others like Snapgene, Pymol, and Rosetta. And those are just to name a few. Those aren't an all-inclusive list. Okay, thank you. And I think we'll get into some questions. I see some have already populated in the chat. Okay.
Yeah.
Um,
Thank you so much, Maya and Leona. Yeah, why don't we dive right into a couple of questions real quick from the chat. So, first things first, from Crystal Candy, what inspired or motivated you in general to explore biology? Like, what got you curious about it?
I mean, I can start with this one. I really liked all the science classes I took, like all the ones in middle school. I had really great teachers in high school. What motivated me, I guess, to explore biology, for example, outside of a classroom was I got really interested in seeing what we were doing like in real life. Like when you learn about a cell or you learn about an organelle or disease, I got really interested in seeing how people like ask questions further beyond like, for example, what I might have been taught in high school. So that motivated me. At least I like did more in high school, like such as doing research to explore biology. But I also wasn't just limited to biology. I liked math a lot. I liked chemistry. I was passionate about various things and didn't specifically just only like biology. So that's what I'll say for that. And I can also pass it on to Leona. She has more about her biology background.
Yeah, so I came into MIT actually being kind of completely undecided in terms of major. When you get into MIT, you don't have to pick a major until, I mean, you actually have till your second year to pick a major. But I always loved science. My main thing in high school was math. I took a lot of college math classes. And I liked how math kind of applied to everything. And I liked the idea of kind of modeling the world with math. And I really liked the idea of doing something that could help people. So I at first envisioned myself maybe doing sort of math and data science and like computational kind of social justice stuff. And I was really into my UNICEF club at high school, raising money for UNICEF. Um, and then I realized that a lot of issues. My computer is about to die. come from kind of health and environmental issues. So I was really passionate about that stuff already. So I came to MIT and I explored other things. I was like super drawn to engineering because this place just makes you love like anything engineering. And so I kind of eventually settled on electrical engineering actually with medical devices. And then I did research in that. And I realized like I want to be working with living things. Like it doesn't make sense to just, I was working on ingestible electronic devices. And it's like even cooler to like engineer the actual biological substrate. And I discovered synthetic biology as a field, like engineering biological circuits, and it has so many amazing applications in environment and in health. And I think it's just going to grow so much as a field. Bioengineering only became a major at MIT, I think, in 2007. And it's just, I think it's going to change the world. So, yeah.
Nice. Yeah. Well said from both of you guys. Um, Another question from Winnie. I guess biology and computer science are two very orthogonal fields. So their question is, how do I level up my coding skills with connections to biology? And how do I better my understanding of which situations might require what solutions? Yeah, I guess it's a very broad answer, but however you guys want to tackle it.
I can chip in a little bit to take a stab at it. I would first say that coding skills in general you can use in any context, and they're not just siloed into like the question you're answering. I started with learning how to program in like my ethical data science research group, and I wasn't even answering questions related to biology, but you can imagine that the skills are can be used to analyzing biological data sets, and the caveat is that there has to be a little bit more research and possibly reading of papers, such as a paper Leona presented of understanding the biological context of a problem. And sometimes that is like needed to supplement as you like think about a project or think about like writing a script that will somehow be used to model like a situation, like a genetic circuit, or be used to like train a machine learning model on data. You have to think more about the biological context of that in terms of understanding which situations require what solutions. I think that sometimes, like in biology, you have two different sides of you want a better sense of a problem, and some things don't exactly have a solution. For example, something you can develop might optimize like how researchers design experiments or optimize like TrackMate does, and what TrackMate Batcher does is how you collect cell tracking information from videos. That didn't have exactly a solution. There's still parameters that need to be tuned. Some researchers don't like TrackMate. There's other plugins they prefer. But what all these things do is they give you like a collection of tools that can help you with the research you're doing. In terms of solutions, s- as I said, that maybe a solution is you want people to have like more accessibility to answering questions, but their doctor isn't available. So that has more of a more structured solution in a sense, because you can think of like maybe designing a bot it can talk to that answers their questions regarding health, that has more of a like shaped solution you can think of, but there's no clear way to defining what situations need a solution in the biological context. And Yona, if you have anything to add, I'll also pass it on to you.
Yeah, Maya gave really, really great advice. She's also the expert here. I haven't, I've done a little bit of computer science. Most of it has just been learned through some experience, like undergraduate research, that kind of thing. I took APCS in high school, but that's like barely experience. But yeah, I really do think it just comes from doing projects and practice. I would still consider myself a beginner. But I think nowadays with the internet, it's super, super easy to learn and practice. And then you have like Claude that can be your personal tutor, basically. Like you can easily search up what the syntax is to do a certain thing. I think, yeah, like once you know one coding language, you can kind of apply it to everything else too. So even if you've never used MATLAB before, for example, if you know Python, like similar concepts, right? Um, and yeah, reading papers helps a lot with understanding what situations connect to what kind of solutions. Um, yeah, I don't, I think over many, many years, people start to just develop an intuition for that kind of thing.
Awesome. No, yeah, that makes a lot of sense. One more question. I think we can wrap it up on this one. Uh, Stepping back, Blueprints, a lot of high schoolers that are coming in with a lot of curiosity. So one question is, are there opportunities that high schoolers can do before going into college? An introduction to like what goes on inside the lab. Anything you guys know about or any insight you can share on that?
So, I didn't do any bio research in high school. I actually didn't do any research in high school at all. I know some people here did. I wouldn't feel any pressure to do that in high school. Like, it's really hard to get those opportunities, and some labs don't allow anyone who hasn't graduated high school in the lab. If you can get the opportunity, it's awesome, but you don't need that. You'll get that in undergrad. But it's awesome that you're super curious. Um, BioMakers is hoping to have some kind of high school outreach program, but that will be in the works for a while because it's hard to get people approved to go into the lab, much less high schoolers. Um, but yeah, like reading papers. And I know papers are really hard to read because it's super dense language. Um, I really recommend kind of just like one paragraph at a time, like writing basically in plain English what was said in each paragraph, or just reading like kind of the at the end, there's like normally a summary of what they did, and then there are all the methods more at the beginning that are way more complicated. Just kind of get the general gist. And yeah, that gives a good introduction to that kind of thing.
And then I think I had a little bit of a different experience just because I was like in a high school in a situation that like allowed me to go into a lab in high school, which is very rare. There are a lot of barriers to, and you can imagine a lot of complexities of having a high schooler in a college lab. A lot of different risks that come up, especially with a high schooler. Um, Lacks sometimes like training skills that are offered to undergrads to be in a lab, and they also have to catch up in sort of the biological context of what's going on in a lab because the high school curriculum prepares you for high school, and there's like some things you'll just never know until you're working on a project, but it's also helpful to, for example, have an undergrad class that gives you some exposure to biochemistry before being in said lab. That being said, as Leona said, that's that's completely rare. I went to an early college that was near a college campus, and that's how I was able to get an introduction to what was happening in a lab. How I knew that I wanted to work in a lab was that I did a bunch of like some science fairs. There's like research being presented at different competitions in my area, and they would like have like with Professor so and so of indicating they worked with a professor in a lab. And I thought, wow, that's pretty cool. I didn't know a high schooler could do that. So I'd say that's at least how I got an intro to being inside a lab. I think it very de like depends on your context, and you might not always live near a college or a lab that's accepting high schoolers. There are exceptions to that where there are a small number of programs that you can apply to, for example, over the summer, where those are designed for high schoolers and to match them with someone to do research with. And those opportunities you can usually find on the web if you look up summer internships or summer lab experiences for high schoolers. And the barrier to that is they can only take so many people. So that's another opportunity, but it's also not available to everyone because you have to get in via application. But I'd say you shouldn't be limited to that. As you can see, both Leona and I are here and both very passionate and involved in labs. We are both in a lab right now, both doing really impactful research. So as Leona said, you can read papers. Like YouTube exists. A lot of researchers now like documenting and showing the work they do, and you can get to walk through that on YouTube. Usually labs all have like a website, so if you have something very specific you're interested in, you can go to like the website of a college, and they'll have like papers or like posters even or like abstracts that were presented at conferences about the work they do. And you can piece all these things together and get more of a sense of what goes on inside a lab, especially if you can't physically be in one. I think at the bottom, at like the end of the day, all you need to be is a curious person. Like you don't need to do all these things, and this is what gets you to be a good researcher, because everyone is still wondering if they're a good researcher today. So I'll end with that.
Awesome. Thank you so much, Maya, Leona, and the entire BioMakers team. For everyone who tuned in, thank you guys so much for coming to the second workshop of Blueprint Week. If you're in the YouTube chat, please go ahead and drop a thank you to Maya, Leona, and BioMakers. And yeah, with that being said, we're going to wrap up this live stream. Thank you guys for tuning in. And be on the lookout for our next live stream coming out tomorrow for our next workshop in the BP Week series. Thank you guys so much. Okay, fire.